Hello, this is part 1 of a 4-part series where I will be breaking down and analyzing the binding pocket of 5-HT2 receptor proteins. Before I get started, you should know I don't explain any structural biology concepts in these videos. These videos assume you already know what proteins are, what amino acids are, and what the various interactions such as hydrogen bonding or pi stacking mean. I also assume you understand various biological concepts such as affinity and the difference between agonists and antagonists. If you are unfamiliar with the terminology used here, I would recommend first watching some videos that cover introductory structural biology. In these videos, I will break down how ligands bind to these proteins and what interactions result in not only the affinity of these ligands to the receptor, but also the selectivity of each one. Finally, I will use the information obtained from this breakdown to demonstrate how we can improve drug design targeting these receptors. I want to preface this by saying these are not published results. This is not some scientific study funded by an institution. This is a personal passion project that I thought I would share on YouTube with others that are also interested in these systems. All of the data, analysis, and conclusions that you see in this video are completely my own, conducted on my own personal computer, unless otherwise stated, in which case I will cite the appropriate study. It should also be noted all the data you will see is from computational models, which without experimental evidence is just that, a theoretical model. However, with that being said, I think a theoretical model can still be useful and helpful in drug design. In this video, I will be analyzing the interactions of antagonists with 5-HT2A and 5-HT2C. In part 2, I will be looking at antagonists at 5-HT2B and comparing them with what we see here. Then I will follow a similar format where I look at agonists at 5-HT2A and 5-HT2C, followed by 5-HT2B. Now, rather than just show a PowerPoint presentation or some pictures from papers, I'll be using a software called Pymol that allows you to visualize these protein ligand structures and interact with them. Now, to explain a little background on this system, what even are these receptors, why do we even care about them? Serotonin receptors are both GPCRs. That includes 5-HT2A and 5-HT2C, which are 7 helical transmembrane proteins that are bound to G proteins. They contain 3 extracellular loops and 3 intracellular loops. They are expressed ubiquitously throughout the body and thus have a variety of roles. However, a lot of the recent focus on these receptors, especially for 5-HT2A and 2C, has been in the brain, specifically the prefrontal cortex. 5-HT2A has roles on mood and memory, whereas 5-HT2C has been shown to have roles on appetite and seizures. For example, 5-HT2A agonists, such as LSD and mescaline, can cause short-term memory complications and mood fluctuations, whereas 5-HT2A antagonists have been used as antidepressants and antipsychotics. 5-HT2C agonists, on the other hand, can cause appetite suppression, and antagonists are currently being looked at as anticonvulsants. Due to a variety of factors, though, many ligands that bind to 5-HT2A, whether agonist or antagonist, can also bind to 5-HT2C as well. This is commonly seen in weight gain problems to those treated with antidepressants. And that's in fact one of the reasons for this video. A better understanding in what contributes to selectivity will help better aid drug design so more selective drugs can be made with less side effects. So let's start with 5-HT2A that I have displayed here. I'll first cover what we know based on previous studies and structural work. In order to understand how 5-HT2A functions and is activated, we must first compare the inactive and active conformations of this protein, which have both been solved in the past. In blue is the inactive conformation bound to methyphenidine, and in white is the active conformation bound to 25CN and BOH. And on the left here, I have a photo from the paper that solved the active conformation that will accompany us as we go through these interactions within Pymol. 5-HT2A, like all other GPCRs, has a similar mechanism of activity. And we will first focus on the rearrangement of the toggle switch tryptophan 336 seen here in the photo and seen here in Pymol. This is accompanied by a rearrangement in the PIF motif, phenylalanine 332 and isoleucine 163, seen here on the left in the photo. This causes the entire helix to shift to the right, which results in a disruption of the ionic lock, which is between the arginine and the dry motif, 173, and the glutamic acid, 318. You can see that here in Pymol as well. Once the helix shifts to the right, this arginine is shifted upwards, and the glutamic acid now shifts downwards to interact with the G protein. There are other rearrangements, such as the NPXXY as well, that shifts to bind to the G protein. However, for the focus of our video, since we will be discussing the binding pocket, I will only be focusing on the toggle switch and PIF motif. In a similar manner, in order to understand how 5-HT2C functions and is activated, we need to compare the inactive and active conformations of this protein as well. In green is the active conformation, and in blue is the inactive conformation of this protein. And we can see, similar to 5-HT2A, we'll start with the toggle switch. You have a less pronounced rearrangement upon activation, accompanied by a rearrangement in the PIF motif, that same phenylalanine and isoleucine. This causes a shift in the helix, again, less pronounced than what we'd seen before, but it results in the disruption of that ionic lock between that arginine and glutamic acid. As you can see here in green, the arginine now shifts upward again, and the glutamic acid now shifts downward to interact with the G protein. This also has similar rearrangements in the NPXXY motif as well, but as before, we only care about the binding pocket, so all I will be focusing on is the toggle switch and PIF motif in 5-HT2C as well. 5-HT2A has two binding pockets. You have the main binding pocket, also known as the orthosteric binding pocket, which resides above the toggle switch and PIF motif. And then you have the extended binding pocket that extends out to helix 3, 2, and 7. Every single ligand that binds to the extended binding pocket still interacts and binds to the orthosteric binding pocket as well. This holds true for 25CN and BOH as well, which, as can be seen here, while its chain extends into a unique binding pose, which I will discuss later when we're discussing the agonists, it still binds and interacts with the orthosteric binding pocket. Now, as you might expect, the same holds true of 5-HT2C. This also has two binding pockets, the main or orthosteric binding pocket that resides above the PIF motif and toggle switch, an extended binding pocket that extends out to helix 2, 3, and 7. Now that we know how these receptors are activated and where the ligands bind, we can start to understand how these antagonists actually work. So we know that the toggle switch and PIF motif have to be rearranged for activity. So what these antagonists do is they bind into this orthosteric binding pocket, interact with these amino acids, and prevent the agonist from binding and activating the receptor. Now, these amino acids themselves can spontaneously rearrange, and that's what results in the intrinsic activity of these receptors. So what inverse agonists do is they bind into the same binding pocket, interact with these receptors, and lock them in place, preventing them from spontaneously rearranging. And this can be seen in all of the structures of the antagonists we have. All of them bind to this orthosteric binding pocket and extend down and interact with the toggle switch in the PIF motif. Now, considering how similar we've seen 5-HT2A and 5-HT2CB so far, it should be no surprise then that antagonists function in the same way at 5-HT2C. They bind in the orthosteric binding pocket and interact with the toggle switch in the PIF motif. Now you may be wondering, if the binding pockets for both these receptors is the same and they function in the exact same manner, what's the difference between them? And this is in fact the big issue here. If we overlay the antagonist bound state of 5-HT2A with 2C, we can see that the ligand binds very similarly, and they have the exact same interactions. And this is why many ligands that bind to 2A also bind to 2C. Now, if we take this a step further and we look at the amino acid composition or the sequence, there is 70% similarity between these two receptors. However, if we take it a step even further and look at just the amino acids in the binding pocket, which I have highlighted in green here, we can see that they're almost identical. There are only two exceptions. Leucine 228 in the extracellular loop 2, which is a valine in 5-HT2C. However, both of these are hydrophobic amino acids. They're structurally very similar. They can partake in the exact same interactions. And serine 242, which is an alanine in 5-HT2C. And there's been a bit of work done on this in the past, where they've mutated this serine to an alanine and seeing how does it affect binding and activity. And what they saw is, well, it did affect LSD's binding a little bit and the activity as well. It doesn't influence other ligands like serotonin and 25CN and BOH. And if we look at the structures here, we can sort of get an understanding of why, right? This serine is not even facing the antagonist, which means the ligand is not actually interacting with this amino acid. And if we look at other ligands, again, the serine is facing away in every single case. So it's not interacting with any of these ligands. And so its mutation should have minimal effect or none. So if it's not the sequence that causes this variation, and it's not how these ligands bind, what causes a change then? How do we have selectivity with these two receptors? And how can we use that information to design drugs better? And that is ultimately the purpose behind this video and the questions that I'm looking to answer. One final thing we also need to cover are three particular interactions that are crucial for binding. While these are not crucial for activity, for example, agonists and antagonists both need to have these interactions, they are critical for the ligand binding to the receptor in the first place. And those interactions are a salt bridge formation with an aspartic acid and pi stacking and hydrophobic effects with phenylalanines 339 and 340. You'll notice that in every single ligand that we cover, both antagonists and agonists, each has an aromatic group that binds in the orthosteric pocket and a nitrogen that is a specific distance away from this aromatic. We see this with antagonists such as methiopeptine up here and MDL 10907 down here. And we have this as well with our agonists. You have serotonin, you have your phenethylamines, and your ergotamine as well. All have this nitrogen within a particular distance of the aromatic. And if you mutate these amino acids, aspartic acid and phenylalanine, you will lose complete binding and activity. So this sort of limits and restricts the compounds that you can design to target these receptors. To better understand what factors contribute to the selectivity of these antagonists, what I have done is I have docked multiple different antagonists, some of them selective, some of them non-selective, to 5-HT2A and 2C. And we're going to go through these docked structures and compare them and see what factors contribute to their selectivity or lack thereof. Starting with the crystal structure of methiothepine, which has similar affinity to both 5-HT2A and 5-HT2C. You can see here in this Excel sheet, I have a list of the pKi values for each of these compounds that I have docked. So if we first look at melloryl, which we can see here is structurally similar to methiothepine, and like methiothepine, is not selective for 2A or 2C. So in cyan here is my docked structure, and we can see if I bring up the toggle switch, the PIF interactions, and the aspartic acid, we can see that it does in fact interact with the toggle switch and PIF motif. And the nitrogen is right where it should be as methiothepine is to interact with the aspartic acid. So it binds similar to methiothepine. Now if I also overlay the 5-HT2C docked structure, we can in fact see that it binds identical to the 5-HT2A docked structure. It has the same interactions down here, and again, the nitrogen is in the proper position to interact with the aspartic acid. So we can see that all these interactions that melloryl has with 5-HT2A and 5-HT2C are similar. And that can explain why it's not really selective for one or the other. Now, if we instead look at mesoridazine, which is an analog of melloryl, where you add a carbonyl here, what you can see is you increase the affinity to 5-HT2A and decrease its affinity to 5-HT2C. So in fact, now you've gone from a non-selective ligand to a selective one. And we can see why this is the case in my docked structures. So here in yellow, we have mesoridazine and 5-HT2A. And we can see that it binds similarly to methiothepine. Again, you have this aromatic that interacts with the toggle switch and the PIF motifs. The nitrogen this time is slightly different in terms of position, but it's still right next to aspartic acid and can form a salt bridge with it. However, what's changed now is the direction of this methyl group here. Now it's pointing towards helix 3, and the carbonyl that we added is pointing towards helix 5. If we look at the docked structure of mesoridazine with 5-HT2C on the other hand, we can see again it binds similarly. You have the aromatic in the right position, and the nitrogen can interact with the aspartic acid, so those criteria are met. However, the carbonyl and the methyl group are pointing in the complete opposite direction this time. Instead, they're pointing towards helix 6. So what this allows mesoridazine to do in 5-HT2A is it can now better interact with leucine 229 up here in the extracellular loop 2. And we know this interaction is critical for the affinity of these ligands to these receptors. And the reason why I suspect this carbonyl causes this methyl group to point in the opposite direction, whereas if we compare to melloryl, we can see in all cases they all point towards helix 6, is I believe because of this kink that you see here between 5-HT2C, which is in the pink color, and 5-HT2A, which is in the white and yellow here. And this kink is in fact caused, if I bring up the sequence here, by a proline. In 5-HT2A, this amino acid is an aspartic acid. However, in 5-HT2C, it's actually a proline. So what this proline does is it causes a kink in helix 5 that results in a shift of the helix to the left that goes down the entire chain. And this results in two changes. One, the orthosteric binding pocket of 5-HT2C is now in fact more open than 5-HT2A, and this can actually change how various ligands bind, as we'll see later down the line. And two, this results in this phenylalanine here in 5-HT2C pointing towards helix 3. And so this might be a better angle to see. So what happens in 5-HT2C is, in fact, this pocket here is blocked. The carbonyl cannot point in that direction. And that's why we see the carbonyl instead pointing towards helix 6. In 5-HT2A, though, you don't have that kink, and so the phenylalanine is pointing towards the intracellular side. And that allows the carbonyl to point to helix 5, resulting in the methyl pointing towards helix 3 and interacting with that leucine 229. And I believe that's what results in the selectivity that we see of mesoridazine between 2A and 2C. Let's look at another antipsychotic now, loxapine. You can see it's structurally similar to methiopepine. And in terms of affinity, it does bind slightly tighter to 5-HT2A. Now, if we look at the docked structure of loxapine, 2,5-HT2A, we can see it does slightly shift to the right, but overall, it does bind relatively similarly. You have your aromatic here interacting with toggle switch and the PIF motif. The chlorine is pointing upwards where the serine and methyl group are here. And you can see your nitrogens here are also relatively similar and can form salt bridges with aspartic acid 155. If we look at the dock structure of loxapine to 5-HT2C, we can see now it's slightly shifted to the left, but it has more or less the same interactions. Again, PIF and toggle switch over here. And it's in position to form a salt bridge with aspartic acid 155. So overall, they do bind relatively similarly. There is a slight shift left to right, and I think that's primarily, again, because of the shift of the helix. Now that it's slightly more open, this can shift slightly more to the left. But again, the similar binding pose is probably why there isn't that big of a deviation between 2A and 2C, as what we saw in, say, for example, mesoridazine. So far, we've discussed 5-HT2A selective antagonists and non-selective antagonists. Let's now shift the focus to 5-HT2C selective antagonists, specifically SDZ-SCR-082, which preferentially binds to 5-HT2C. Structurally, you can see here it's quite distinct from methiopepine, but it does have that aromatic and that nitrogen that are in the proper position to interact with that aspartic acid. So let's look at the dock structure of this compound to 5-HT2C first. Here we can see that the aromatic lines up perfectly with the one in methiopepine, interacts with the toggle switch and the PIF motif. And we can see here that the nitrogen is also in the proper position to interact with the aspartic acid. Now, while we've seen bulky compounds, this is actually quite different. All the previous compounds have been relatively linear. However, in this case, the pyrroline is instead pointing towards helix 5. If we look at the same compound docked to 5-HT2A, we can see that it instead binds way up here. This is generally where agonists bind, such as LSD. It does not interact with the toggle switch or the PIF motifs. And I think the reasoning why it's unable to get down into this binding pocket is because it's just too small. As stated previously, this kink caused by this proline results in the binding pocket for 5-HT2C to be bigger, which is why I think this compound that's a little bit more bulkier and spread out is able to fit down into this pocket to interact with this toggle switch and PIF motif, whereas the same compound in 5-HT2A is not. In fact, I wouldn't be surprised if this compound is a partial agonist to 5-HT2A, but unfortunately, I could not find any studies where they looked at the activity of this compound to this receptor. Let's look at one final compound, prolixin. This is like methiopeptine, and if you may notice, it's actually very similar to meloril, except it has this hydroxyl extension over here, which we'll see the importance of later. And we can see that it binds more preferentially to 5-HTA over 5-HT2C, which is quite interesting, because as we saw in meloril, there isn't much selectivity going on. So something about this OH group that's added with this ethyl group causes an increased selectivity in 5-HT2A. Now, if we put here the dot structure of prolixin bound to 5-HT2A, we can see that it binds quite similar to methiopeptine. Again, you have the proper interactions here with the toggle switch, and the nitrogen here is in the proper position to form a salt bridge with aspartic acid. And now you have this OH that extends into the extended binding pocket. And if we look at this prolixin, this time bound to 5-HT2C, slight shift to the left. However, again, a very similar binding pose, interacts with the toggle switch. Nitrogen is in the proper position to interact with aspartic acid. However, if we get rid of methiopeptine, you can see how similar this compound binds to these two receptors. They're almost identical outside of this slight shift, with the exception of this hydroxyl group. In 5-HT2A, it's pointing up, and 5-HT2C is pointing down. And this single interaction is actually quite critical for the selectivity of this compound. This hydroxyl group forms a hydrogen bonding interaction with this tyrosine 130, what is this, 6, 7, 8, 9, which is 118 in 5-HT2C. However, we can see that in 5-HT2C, it's instead shifted away towards helix 7. And it no longer is able to form that hydrogen bond with this hydroxyl, which is why this in 5-HT2C, this compound's hydroxyl is pointing down. So the question is, why is this pointing away? And if we look at asparagine 363 and 5-HT2A, this actually forms a hydrogen bond with that tyrosine, as we can see here. However, if we look at the same asparagine, which is now 351 and 5-HT2C, the nitrogen is pointing in the wrong direction. Therefore, it can no longer form that hydrogen bond and bring this tyrosine down to interact with that hydroxyl. So the question is, why is this pointing upwards instead? Well, there is a glutamic acid 347 in 5-HT2C that is actually a glycine in 5-HT2A. And this glutamic acid forms a hydrogen bonding network with this asparagine. So it prevents the interaction with that tyrosine and therefore does not bring it down to interact with that hydroxyl. And that is why you have that selectivity between 5-HT2A and 2C in prolixin. As we will come to see, this network between this asparagine, glutamic acid, and tyrosine plays a critical factor in the selectivity of antagonists that bind to the extended binding pocket. But to wrap up the orthosteric binding pocket, we can see that the difference in the proline to the histidine causes a kink in this helix, resulting in a more open binding pocket in 5-HT2C versus 5-HT2A. And so more bulkier compounds will be able to go deeper into the binding pocket and interact with the toggle switch and PIF motif, which is why I think if you look across the boards, antagonists that bind primarily to the orthosteric binding pocket are not as selective as antagonists that also bind in the extended binding pocket, as can be seen between Mellaril and Prolixin, where the main deviation is just this trifluoro group and this ethanol group that we see here. Now let's move on to the extended binding pocket. And we're going to be looking at two structures for this. The first is 5-HT2A bound to the antagonist risperidone in green. And we can see here it shares the same characteristics of all the other antagonists we've seen. It interacts with the toggle switch and PIF motif. It is in position to form a salt bridge with the aspartic acid. And here we can see it extends into the extended binding pocket between helix 3 and 7. And here in blue we have 5-HT2C bound to the antagonist ritonestrin. And we can see here again it binds to the toggle switch and PIF motif, is in the correct position to form that salt bridge, and it too extends into the same extended binding pocket. Now it's interesting when looking at these structures how similar they actually are, right? They dip down approximately in the same position, they interact with that aspartic acid in the same position, and they bind to the extended binding pocket in relatively the same position. However, we can see here that risperidone is selective for 5-HT2A, where ritonestrin is not selective for either receptor. We can see here that the risperidone does not bind in the exact same way as the ritonestrin. It's slightly towards the left. In other words, it's slightly towards pointing helix 3, whereas the ritonestrin is pointing towards helix 7. Now, there is some evidence suggesting in the extended binding pocket that there is a difference between the activity of the compound in which helix it's pointing to in the extended binding pocket. Here in a paper on 5-HT2B, they looked at LSD and glyceride. And what they saw is glyceride pointed towards helix 3, whereas LSD pointed towards helix 7. Based off these structures and their mutagenesis studies, the paper concluded that antagonists point towards helix 3 and agonists point towards helix 7. However, I don't think this mechanism would apply to 5-HTA or 2C. For one, glyceride is a partial agonist to both of these receptors. And for two, we can see here in the case of one antagonist pointing towards helix 7 and the other pointing towards helix 3, they are both still antagonists. So I don't think this mechanism of one helix versus the other for activity readily applies to 5-HTA2A and 5-HT2C. However, I do think this shift towards helix 3 versus 7 does play a role in selectivity, as I will show in the various structures that I have for these antagonists. Now one might think, why would one antagonist bind slightly towards one helix versus the other in these two receptors? Could it be that it has unique interactions in one helix versus the other in 2A versus 2C? As I stated earlier, the interactions that these antagonists have in the extended binding pocket and the orthosteric binding pocket are largely conserved, as you can see here in green. So it's not that they particularly have any new interactions. Rather, as I stated earlier with the Prolixin, I think it has to do with the hydrogen bonding network that we saw with the tyrosine, the glutamic acid, and the asparagine. And to visualize this, I think a good example is to first see how does the extended binding pocket look before a ligand binds. To visualize this, I will be using the structure of 5-HT2A bound to methiothepine, since that binds only in the orthosteric binding pocket. Now, unfortunately, there is no structure of 5-HT2C where it's not bound to something in the extended binding pocket. So I will be using my docked structure of melaril bound to 5-HT2C, since that primarily binds to the orthosteric binding pocket. And so 5-HT2A is in pink here, and 5-HT2C is in salmon. And if I show the tyrosine here with the asparagine and glutamic acid, what you'll see is there is a difference in this tyrosine here. In 5-HT2C, it sort of occupies the space between the two helices, and that is because it's part of this hydrogen bonding network we had discussed earlier that I can show here. You can see glutamic acid, asparagine, and tyrosine are part of it. Whereas because this glutamic acid is no longer here, the tyrosine asparagine instead shift back to here and form their own interaction further back. What this basically means is that this pocket between helix 3 and 7 is closed off in 5-HT2C, whereas it's somewhat open in 5-HT2A. And we'll see this actually influences ligands binding towards 2C versus 2A. Just to show an example based off of these structures that we have already looked at, if I bring in the 5-HT2A and T structures that we were just looking at, and I can get rid of the methiothepine and melaril structures, we can see here that we sort of have the same thing, of course, to a lesser extent, where in 5-HT2C, this tyrosine is shifted more into that pocket, whereas in 5-HT2A, it's shifted outwards. Same can be said of the asparagine and glutamic acid here, where they are pointing towards that pocket, whereas in 2A they are pointing away. And again, you'll come to see in the various dock structures that this difference between binding sort of towards one helix for the other does follow the pattern of selectivity for both 2A and 2C. So now let's look at one of the docked structures, starting with the selective antagonist MDL 100907. That's commonly used in a variety of studies. So you can see here that it's selective for 5-HT2A, and you can see the structure up here. Now I'm not going to display the PIF motif and toggle switch anymore, but you can see that it does extend down and interact with them here. And I'm not going to show the aspartic acid anymore, but you can see the nitrogen is in the proper position to form a salt bridge with it. Instead, I want to focus on the extended binding pocket. So we can see here that this is sort of pointing towards helix 3. If I overlay the 5-HT2C docked structure of this compound, what we'll see is, everything in the orthosteric binding pocket is basically identical. This hydroxyl is even pointing in the same direction. However, it's this aromatic group with these two methoxy groups that are different. In 5-HT2C, it's shifted towards helix 7, whereas in 5-HT2A, which is in blue, it's shifted to helix 3. And to understand why this is the case, we'll have to look at, again, that network of the tyrosine with the asparagine and the glutamic acid. As we can see here, in white, that the tyrosine does form that hydrogen bond again with the asparagine, blocking off this pocket here, and preventing this compound in 5-HT2C from, from the methoxy going down and pointing into it. However, in 2A, it's instead shifted away, and this methoxy can now point into that pocket. If we look at the 2C dock structure, we can see instead that same methoxy is now forming hydrophobic contacts with the glutamic acid. In fact, this model correlates relatively well to previous structure-activity relationship studies that have been done. For example, in studies where they modified this compound at various positions, what they noticed is they were able to modify this meta position, this methoxy right here, to a variety of different things, and it was well tolerated in 2A, but not 2C. So you can see the affinities here, for example, in 4, where they changed it to a hydroxy group. They saw that the selectivity actually decreased between 2A and 2C. And this might be because without this interaction with the glutamic acid, it might be able to actually shift a little further down, closer to the binding pocket that the 5-HT2A compound is in. But when they looked at, say, 5A and 5B, where they added these big bulky chains, it completely abolished binding to 5-HT2C without altering the binding to 5-HT2A that much. And we can see why that is the case here, right? If you modify this position and add a big bulky group, it has quite a bit of room to extend outward. Whereas in 2C, it's binding to helix 7. So if you add a big bulky group, it has no room to go anywhere, and you're basically abolishing binding. And this is why I think this compound is selective for 2A versus 2C. It's not, this orthosteric binding portion, but entirely because of this extended binding portion. Let's look at another 5-HT2A selective antagonist, ketoserin. I apologize if I'm butchering all of these names, by the way. I've never actually heard any of these compounds pronounced. Again, we can see it's pointing down into the toggle switch and PIF motif, nitrogens in the proper position to form that salt bridge. And so we can see here that the portion that goes into the extended binding pocket is again pointing towards helix 3. And as you would expect, if we take the same compound, dock it to 5-HT2C, we can see it's instead pointing to helix 7. And again, the rest of the orthosteric binding pocket is completely identical. So the selectivity of this compound, again, comes from this extended binding portion. And if I show that tyrosine, asparagine, and glutamic acid portion, here we can see again the tyrosine is for 5-HT2C is shifted a little left into the pocket, whereas in 2A it shifts further outward. We can see here that the aromatic ring has hydrophobic interactions with the glutamic acid, and there is an additional factor that I have not previously discussed, and that is this lysine, which is an alanine in 5-HT2A. And we can see it's actually a part of this hydrogen bonding network over here between the asparagine and glutamic acid. And it actually enables the extended portion of ketoserin to shift even further towards helix 7 than we had seen in MDL 10-0907, for example. So this is, again, another factor where the more selective compound is pointing towards helix 3. And this is exactly what we had seen in the case of MDL 10-0907 as well. The idea so far has been that glutamic acid in 5-HT2C is what's causing all these rearrangements in selectivity between these compounds. So the next step is let's mutate that to a glycine and see if we can dock the compound similar to what would be seen in 5-HT2A. And that's exactly what I've done here in pink. We can see the orthosteric binding pocket is identical, which is to be expected. And we can see that there is actually a change in the extended binding pocket. While it does shift slightly to the left, it doesn't shift as far as the same compound in 5-HT2A. What's interesting to see is instead of that tyrosine shifting backwards, it instead shifts forwards and interacts directly with the nitrogen in the ring here. And as would be expected, though, the lysine and the asparagine over here, if I can get it to show, have shifted all to the right since they're no longer being held by that glutamic acid. While this result is a bit disappointing, I do think it highlights an important example that all of this is just a theoretical model, right? I do still think that that glutamic acid does have a role in selectivity, but without empirical evidence, this is all we sort of have to work with here. And sometimes the model that we get doesn't always align well with what we would expect. So let's look at another 5-HT2A selective antagonist, this time spiperone. And we can see here in gray, it's bound to 5-HT2A, and again, dips down, interacts with that PIF motif and toggle switch. You have a nitrogen here that can form a salt bridge with the aspartic acid. And we can see here that the portion that goes into the extended binding pocket points towards helix 3. If we overlay the 5-HT2C docked structure, we can see something a little interesting. We can see down here instead you have it shifting slightly to the left. It can still interact with that toggle switch and PIF motif. However, this shift to the left pulls it a little slightly back. And instead, we can see it points towards helix 7, but it doesn't go as far into it as the other compounds we've seen so far. And this slight shift to the left is again possible because the orthosteric binding pocket of 5-HT2C is slightly bigger than 5-HT2A, as we saw previously. However, if we again look at the tyrosine and amino acids involved within that network, we can see the same pattern, right? We can see here in 5-HT2A that it points towards helix 3, and then in 5-HT2C, that tyrosine is coming in and blocking it off, preventing this aromatic here to point in that direction, right? Otherwise, you would get a steric clash. And again, that is a result of this hydrogen bonding network between that glutamic acid, the asparagine, and the tyrosine, as you can see here, which forces this to point towards helix 7. And this actually correlates relatively well to structure-activity relationship studies that have been done in the past. If we look here, by itself, this compound does not readily bind to 5-HT2A or 2C. They say 1C, but keep in mind 2C used to be 1C, so it's the same thing here. However, we can see that once you add this big bulky group over here and can actually go and extend into the extended binding pocket, you start increasing the affinity of this compound for these receptors. And you can see it is somewhat length dependent. As you decrease the length, it doesn't extend as readily out, and therefore your affinity starts to decrease more and more. If we start comparing this fluorine to the methyl group, or the nitrile group here, we can see why you get the reduced affinities here, right? This phenyl group is pointing towards this tyrosine, the methoxy or nitro group might be too big to the point that it actually extends a little too far out and causes some steric problems with helix 3. And so that's why adding a smaller hydrophobic group like a fluorine would be the best case here. So we've seen in 3 different examples here that antagonists that extend towards helix 3 or closer to that side are in fact more selective to 5-HT2A than 2C. However, we will actually see that the same is also true for 5-HT2C. So we've seen selective 5-HT2A antagonists. Now let's start looking at some selective 5-HT2C antagonists. Starting with SB242084, which you can see here selectively binds to 5-HT2C. Now, if we look at the orthosteric binding pocket, we can see that this chlorine and this methyl group, along with this aromatic, interact with the toggle switch and the PIF motif. This nitrogen can interact with that aspartic acid. And in the extended binding pocket, we see something different now. We see that this aromatic with this methyl group is actually pointing towards helix 3 instead of what we've seen previously where these compounds point towards helix 7 in 5-HT2C. And this tyrosine, instead of being part of this hydrogen bonding network with the glutamic acid and asparagine, is instead pointing down and forming a hydrogen bond with the nitrogen in this group. So this is quite different from what we've seen before of the 5-HT2A selective antagonists. Now, if we add the 5-HT2A docked structure, what we can see is this does not actually come down into that binding pocket and interact with that toggle switch and PIF motif. And this is a result of, again, the shallower binding pocket for 5-HT2A versus 5-HT2C, which allows it to sort of basically shift a little to the left and go deeper to that left than in 5-HT2A. If we look at the extended binding pocket, we can see as a result it does extend further out towards helix 3, and we can see that tyrosine's shifted away to accommodate it. However, you can see that in this case, unlike the other scenarios, the position in terms of what they're pointing towards is relatively similar. So we can see, while I don't think 5-HT2A and 2C are like 5-HT2B, where the antagonist pointing towards helix 3 makes it an antagonist versus pointing towards helix 7 makes it an agonist. I think in this case, though, that the selective antagonists point towards helix 3, whereas the non-selective ones point towards helix 7, as we had seen in the case of ridotserin in the 5-HT2C structure, which was pointing towards helix 7. Finally, let's look at one last 5-HT2C selective antagonist, RS102221, which we can see here selectively binds to 5-HT2C. If we see here, it interacts with the toggle switch PIF motif, it can interact with the aspartic acid here. And if we look at the extended binding pocket, we see something interesting. It's pointing towards helix 3, and if we look at the tyrosine, instead of pointing down into that pocket, it's instead shifted upwards to form that same hydrogen bonding network, this time with the lysine and asparagine here, which is quite different from what we'd seen with the 5-HT2A selective antagonists, but what we'd seen in the 5-HT2C selective antagonist, SB242084. Now, if I add the 5-HT2A docked structure, we can see here it also reaches down and interacts with the toggle switch and PIF motif. However, there is a difference in this upper portion over here, where in 5-HT2C, this portion sort of gears towards helix 2 and is shifted to the left, and 5-HT2A goes straight up. Again, this is because the orthosteric binding pocket of 5-HT2C, especially over here, is bigger than 5-HT2A. If we continue it along, we see it actually binds in the extended binding pocket identically. So what makes this one selective then? Well, in this case, because of this shifting towards the left, it has a unique interaction with a series 239 over here. Additionally, you can also get a unique interaction here with threonine 139 with the oxygens over here as well. So this gives this compound a unique interaction here with helix 3 that enables it to bind selectively to 5-HT2C. But we can also extend this by looking at the extended binding pocket, pun intended, where we can see that same pattern of selectivity where the extended portion is binding towards helix 2. So this is similar to what was seen in SB242084, and this is also exactly what was seen in all the 5-HT2A selective antagonists as well. And so again, I think this is what determines selectivity in the extended binding pocket between the compounds at 5-HT2A versus 5-HT2C. Finally, there's one other factor that I also wanted to cover, and that is affinity. So not only can we design more selective compounds, but we can also design compounds that bind tighter to the receptor. And here I am basically showing the affinities of everything we've covered, and I've split it into two sections. You have the antagonists that bind primarily to the orthosteric binding pocket, and here are the antagonists that can also bind to the extended binding pocket. Now I have overlaid all the antagonists that bind exclusively to the orthosteric binding pocket here, and you can see that they bind primarily more or less the same. And if we look over here, you can again see outside of prolixin, which does go into the extended binding pocket, they all bind relatively the same. And even if we look at these additions here, where some of them add sulfur groups and some of them add chlorine groups, they all, once again, bind more or less in the same direction, except for the mesoridazine. And we can see here that this explains why these compounds are more or less not that selective for 5-HT2A versus 2C. They all basically bind in the same way. And this is, as I stated before, because almost all of the amino acids that interact with these compounds within the orthosteric binding pocket are conserved. Now, there are a few things we've seen we can manipulate. We can make the compounds bulkier horizontally rather than vertically, as seen here, to take advantage of the more open binding pocket of 5-HT2C than 2A. Or we can add some type of bulky group up top over here, such as this carbonyl, that takes advantage of that phenylalanine that had shifted due to the introduction of this kink from that non-conserved proline. Now, if we look at the affinities of the antagonists that bind to the extended binding pocket, we can see that these are far more selective than the compounds that only bind to the orthosteric binding pocket. And so if I remove some of these compounds that only bind to the orthosteric binding pocket, and I introduce some of these compounds that can also bind to the extended binding pocket, we can see here that they more or less bind in the relative same position, all sort of towards this helix 2 that we've been discussing throughout this video. And something that is of interest is you can see that in the orthosteric binding pocket, they can take a variety of different shapes and forms. Yet all of them are selective, and all of them can bind with just as much affinity as the ones that only bind in the orthosteric binding pocket. So I think both in terms of selectivity and affinity, the focus for designing these compounds should be less on the orthosteric binding pocket spot and more on the extended binding portion. There's one other factor I also wanted to discuss here, and that is if we look at the structure of these compounds. Let me bring this up and this down so you can see everything. Here we have our compounds that bind primarily to the orthosteric binding pocket, and here we have some compounds that bind to the extended binding pocket. So this is risperidone, this is MDL 100907, this is spipirone, and ketaserin over here. Now if you look at the extended portion for each of these compounds, you'll notice there is an oxygen in each case. However, I specifically wanted to focus on the oxygen that is right before that extended portion, such as here in MDL 100907 and here in spipirone. Now if we look at these docked structures, we can see in the MDL structure that it actually points and interacts with the extracellular loop 2. And while in spipirone, this carbonyl is pointing the wrong direction, in a variety of other docked structures, it's in fact pointing and interacting with that extracellular loop 2, which would result in it binding with a higher affinity. And in fact, this does go in line with structure-activity relationship studies that have been done. If we look at, in the case of spipirone, let me zoom in right here a little bit. You can see in the cases where they add either a carbonyl or an oxygen down here, they get higher affinity than you would if you didn't have an oxygen, if you just replace that with a carbon. You can also see that adding a carbonyl rather than an alcohol does also increase your affinity. This has also been seen in SAR studies done on MDL. Again, if I zoom in over here, move this to the right, we can see if we compare 10 and 16, where the difference is the alcohol versus the carbonyl is more effective than the alcohol group, which does correlate with the SAR study of this compound as well. So I'd also say in these extended binding pockets for these antagonists, it's good to add a oxygen here, preferably a carbonyl, right before the portion that actually goes into the binding pocket. So what is the point of all of this at the end of the day? Why are we going through and covering how these antagonists bind to their respective receptors? Well, the whole goal is in understanding what makes these compounds selective. We can then use that information to design more selective antagonists that bind to their respective receptors even more selectively than before. And that's really the whole point of the video. So to wrap things up, I've designed a couple of my own compounds here based off the information that is obtained from this video and from these docked structures. And I will format it into two sections. In one, we will be introducing selectivity to the compound, and in the other, we will be removing or breaking the selectivity of that compound. So to begin with, I want to look at compounds that bind in the extended binding pocket. Now it's important in compounds that have these bulkier groups and these aromatic rings that they point in between the helici of 2 and 7. And that's because, as we've discussed before, that rotation of that tyrosine with the hydrogen bonding network of the asparagine and glutamic acid in 5-HT2C is what results in the selectivity of these compounds. And every compound binds to its selective receptor in this pocket pointing towards helix 2. And so if we take this ring and we instead make it flat so that it doesn't actually dip into this pocket here between these two helices, what you'll see is you actually lose selectivity. And so that's what I've done here. You can see in green, this is 5-HT2A. And all I've done is I've taken this ring and I basically switched these positions around, where now it's flat against helix 2 and 7 instead of pointing into the pocket between them. And if I also show the dock structure of 5-HT2C, let me remove the keraserin so it's easier to see, you can see that they bind basically identically. I have now removed the selectivity of this compound, and it can bind to 5-HT2A and 5-HT2C identically because it is no longer interacting with this tyrosine, asparagine, glutamic acid hydrogen bonding network. So the next thing I wanted to do is get a compound that is selective to 2A and make it even more selective. And so to do that, I will be using the compound M100907. And this compound, as we discussed before, selectively binds to 5-HT2A. And we saw earlier in SAR studies that you can change this 2 position, and while you'll increase its affinity to 2A, you will destroy its affinity to 2C, because as we discussed, there's room here to expand, but in 2C you're pointing towards helix 7, and so you'll no longer be able to bind if you make a big bulky addition here. But instead what I wanted to do is actually fortify or strengthen this interaction of this extended portion to helix 7, and inversely strengthen the interaction of this same position to helix 2 in 5-HT2A. To do that, what I did was modify this methoxy group, which has hydrophobic interactions with phenylalanine 339 and 340, which we know are important for the binding of these compounds to these receptors, to something more hydrophobic. So I changed it to an iodine. And here, if I show the docked structure of this compound, we can see it does bind very similarly to the same compound in 5-HT2C. However, the difference there, let me get rid of 2A as an example. When I dock these compounds, I usually look at various different conformations that the compound can adopt to try and get an understanding of how these compounds fit within these binding pockets. For most of the situations, these compounds can actually adopt multiple different conformations. What I'm displaying here and showing to you guys are the ones that are the most reasonable and energetically favorable, and those that make sense. For example, the ones that form the salt bridge with the aspartic acid and interact with the toggle switch and PIF motif. Now, normally you get, again, a variety of different conformations. However, in the case of this iodine variant, out of the 20 dock structures that I got for this iodine variant, they all bound in the exact same way. Effectively, what this modification does is it locks this compound into this conformation. And so it basically strengthens the interaction that it has with helix 7 and that phenylalanine 339 and 340. Now, if we take the same iodine variant and we look at it docked to 5-HT2A, we can instead see, let me get rid of the 5-HT2C structure there. We can see now that the iodine is pointing instead towards helix 2, and the methoxy, as before, is pointing towards this pocket. All right, so I'll overlay the 2A structure here so you can sort of get an idea of what's going on. And in fact, this iodine now forms a unique interaction. Let me get rid of this, with a serine 131 and 5-HT2A. So effectively what I've done by adding this iodine is I have strengthened the interaction of 2C with helix 7, and I have increased the strength of the interaction of this compound in 2A with helix 2. Right? And as we said earlier, there does seem to be a difference potentially in functionality and selectivity between which helix you're interacting with or pointing towards. Now let's do the reverse, right? We have, in this case, increased the selectivity. Let's completely abolish it. And to do that, what I will be doing is changing the position of this methoxy from a 2 to a 4 and 5. All right, so let's get rid of these and add this example here. So in this example, what I've done is instead of a 2,3 methoxy substitution, I've done a 2,5 methoxy substitution. And if I overlay the 5-HT2C structure of this compound, we can see that we've effectively eliminated the selectivity of this compound. They now both bind in the same position. By moving this 3-methoxy, what you've effectively done is you've disrupted its interaction with this glutamic acid, and so now it's free to rotate and interact with helix 2. The same can be true, of course, if we change this 5-methoxy instead to a 4-methoxy. So if I get rid of these two and I now change it here, so now you have a 2,4-methoxy. You can see in the 2A docked structure, it's relatively similar to what you had previously in the 2,3 docked structure. However, in the case of 5-HT2C, it now docks identical to 5-HT2A. And that's because, again, by removing the 2-methoxy, you disrupt that interaction with that glutamic acid, and now you effectively enable it to point into this pocket between helix 2 and 3 and move that tyrosine. So they effectively bind the same, and you have, once again, lost selectivity of this compound. Finally, I wanted to look at modifications that we can apply to the orthosteric pocket a little bit. Because while I stated earlier, I think more focus should be made on trying to make modifications for the extended pocket, there are stuff we can do for the orthosteric binding pocket as well. For example, if we look at the selective antagonist SB242084, we saw earlier that this group here can extend further to the left, allowing it to shift deeper into that pocket with the toggle switch and PIF motif compared to 2A. And that's effectively what makes this compound selective for 5-HT2C. But let's first disrupt this selectivity. So the problem is the compound cannot extend all the way down. So one thing we can do is actually add a bulkier group here to get rid of that selectivity, which is effectively what I've done here. Now I'm not going to have a diagram anymore over here on the left showing the structure, because I think these are pretty self-explanatory modifications. So we can see here I have instead removed the chlorine and methyl group and added a ring. And now we can see that, in fact, 5-HT2A can come further down. It can slightly go to the left and dip down, interacting with that toggle switch in PIF motif. So one of the modifications is, if you make a bulkier group, perhaps you can make a bulky group and add a chain that goes down so that the bulky group's points and binds further to the left here, where you can do that in 5-HT2C, but not 5-HT2A. Now, the next thing I wanted to do is take this compound where we had basically destroyed its selectivity and reintroduce that selectivity, but makes a modification manipulating the differences between 2A and 2C. So if I get rid of these guys and introduce this case here, where you have your 3 rings that you had in the old scenario. Now I have added this sort of bulky 6-membered ring over here in the corner. So what this results in is, because again, this binding pocket down here is a little bit closed off, it forces this ring to point upwards, and again point out of the PIF motif and toggle switch. In 5-HT2C, we can see that adding this bulky 6-membered ring is not a problem because, again, in 2C, that orthosteric binding pocket is bigger and can accommodate it. So you can see here you still do have that interaction with the PIF motif and toggle switch, whereas you no longer have it as effectively in 2A. So we've made this compound now 5-HT2C selective once again. It should be noted that that size of the bulkiness is important. For example, if we make a smaller modification, so instead we now add a 5-membered ring, we can see that in 5-HT2A, actually let me bring back the old one, you can see it's able to once again dip down and interact with that toggle switch and PIF motif. And if I add 5-HT2C, you can sort of see the same thing. So we've once again eliminated the selectivity of this compound. So the size of the bulky group that you add is important. The bigger it is, a 6-membered ring, maybe a 7 or 8-membered ring, the more selective it'll be to 2C than 2A because of that bigger binding pocket in 2C. Finally, I wanted to take this moment and state that this is of course not all-inclusive. There are many things that I wanted to cover in this video that just for the sake of time, I couldn't really get into. For example, if we look at the structure of 5-HT2A methiothepine, I have largely ignored discussing the role of this serine 242 and this threonine 160. Now this is in part because in none of my antagonistic structures did I see any interactions with serine 242. However, it should be noted that these two amino acids do in fact form a hydrogen bond here. And this may be part of the reason why the binding pocket here is a little more closed off in 2A than 2C, right? This interaction brings these two helices together. What it could also do is it could prevent 2C, or it could, prevent this threonine 160 from interacting with the ligand. Now it's been seen in other serotonin receptors, for example here 1HT1B, this threonine actually interacts with the receptor. You can see here, for example, the threonine is hydrogen bonding with that nitrogen. And in fact, in 5-HT2B, I can show a photo here, it was also shown to interact with the agonist. And mutation of this threonine prevented the activity or the agonism of that compound. So it can be seen that the interactions of these threonine with those compounds are quite important. And it might be that the serine, in fact, interacts with this threonine, preventing it from interacting with the compound. It should be noted that all of these examples, though, are of the agonist-bound states. So it might be that this threonine doesn't actually have a role with antagonists, which is why I didn't really cover it. Another factor that I think is important that needs to be brought up is sometimes you may have noticed some of my structures were labeled with a 6A93. And that's a different PDB structure that I had used to dock. Because the starting structure that you use can actually determine the docked structure that you end up getting. An example of this, for example, is the difference between the LSD-bound state and the methiothepine-bound state. Both of them represent the inactive conformation. But you can see this helix 5 over here and helix 3 both go more into the orthosteric binding pocket. What that means is that in the LSD-bound state, the receptor's orthosteric binding pocket is smaller than in the methiothepine-bound state. And this causes quite a variety of differences when you dock your compounds, because as we saw, the size of that binding pocket in 5-HT2A versus 5-HT2C can cause a variety of differences. And so while I didn't really discuss it or go into it, in multiple cases I have docked the compound to all the various 5-HT2A structures we have to make sure that the results are consistent across the board. I also, for example, have not covered anything regarding 5-HT2A homo or hetero dimerization. For example, there have been computational studies done, let me zoom in on this, where it's been shown that this helix 4 is the site of heterodimerization with mGlu2. And this only occurs, based off these studies, in 5-HT2A, not 5-HT2C. Now, as we saw, some of the 5-HT2C selective antagonists do end up binding into this orthosteric binding pocket more to the left and may actually interact or shift this helix 4, which may have some roles in influencing whether it can or cannot end up dimerizing with mGlu2. However, this is way beyond the scope of my docking studies, but it is something also interesting to think about, which I didn't really discuss as well throughout this video. And finally, I think it's important to also state that this is, again, all a theoretical model based off of computational docked structures, right? And these are not always accurate. One example that I just wanted to go through is Haldol, which is selective to 5-HT2A. And here I am showing the overlay of the dock structure in 5-HT2A and 5-HT2C, which is in green. You can see in the orthosteric binding pocket, which as we expect is identical. They bind to the toggle switch and pif motif, and we would expect that the variation would be in the extended binding pocket for this compound, where in 2A you bind to the left towards helix 3 and helix 2, and in the other case, in 2C, you would bind to the right where you're pointing towards helix 7. Unfortunately, you don't really see that be the case here. Yes, in 2C, it is slightly binding more to the right. However, if you look at the tyrosine and that hydrogen bonding network with the asparagine, lysine, and glutamic acid, they are pretty similar, and I really can't find a reason to explain why this compound is selective to 5-HT2A. Now, rather than think that the entire model and all the docked structures I've stated earlier and this pointing towards one pocket or the other is just completely wrong, I think in this case, the dock structure just this is not a good model or representation potentially of how this molecule docks to these receptors. And so again, at the end of the day, it's important to note that this is all just a theoretical model. There is no evidence that I have seen, experimental evidence, for the role of this glutamic, lysine, tyrosine, hydrogen bond, and asparagine in 2C in determining selectivity of these compounds, which is why I made the video. I think these are unique interactions that determine the selectivity of these compounds that have not been looked at before. Anyways, I'll leave it at that. I think this video is already long enough. I hope it's been entertaining. I hope it's been interesting. I hope you've learned something new. I plan to do this sort of in-depth look next at 5-HT2B and antagonists that target that receptor and trying to determine why and how you get selectivity there. And then after that, I plan to look at agonists at all three of these receptors in sort of the same manner and determining how you get selectivity there as well.